Hello everyone, this is ChemBioSpark. I'm gonna discuss about pharmacophores in this video, and I'm gonna compare different aspects of conventional and traditional drug design approaches with today drug design approaches. And I prepared some structures here, and they share a common uh, uh, pyridine uh, functional group attached to a CF3 moiety, and the entire moiety is observed in these 5 different structures. And the first thing I want to say is that this motif, which is colored in red, is observed in 4 different therapeutic regimens. And so in the first structure, tipronabir is a protease inhibitor of HIV, and taranabant is a ligand molecule for cannabinoid system of the brain, which is responsible for obesity, for actually the satiety process, and our willingness to eat, and valneprid, another ligand for cannabinoid system in the brain. The fourth structure is SB705498, which has reached the phase 2 clinical trial for treatment of rhinitis and chronic cough. And this one is, its mechanism of action is somehow unknown, but it is thought to some degree is mediated by the inhibition of TRPV1 inhibition and antagonism. And the final structure is foloprigon, which is a fungicide and an inhibitor of succinate dehydrogenase enzyme. And all of these structures share this common moiety, this common scaffold, and what we have different variety of actions for these molecules. Although this pharmacophore is not very large and it is very small, and so it is logical to speculate that this moiety can have different consequences when it is fused to a large entity, and this larger entity would therefore guide the therapeutic response of the whole molecule. So the first thing to note is that the other parts of the molecule is also important. So in traditional drug design, when researchers try to manipulate the structures and obtain better structures with more potent, I should say, therapeutic responses, they encounter some molecules which display different physiological and pharmacological responses. So these different ligand molecules were obtained from those studies, and the second thing to note is that the newer drug design approaches are using the energy values for determining whether a molecule is suitable for a protein macromolecule or not, because it is hard to predict based on 2D dimensional structures and without knowing anything about the protein structure, without knowing which part of the molecule is responsible for the most pharmacological response, which part is mainly involved in the inhibition or activation of that enzyme. And so based on this fact, we now use in silico techniques and molecular docking, for example, or molecular dynamics for considering the energy values of those molecules. And the third thing I want to say that is even when you use a large pharmacophore and your structure you think is really selective for that molecule, and you are manipulating that structure to yield better compounds, you should keep in your mind that your final structure might not be selective, and this should be assessed whether by in silico techniques or whether by in vitro techniques, either which you prefer, and of course by the cost effective nature of the computational approaches, you might find it more appropriate to use them. But for many cases, a large pharmacophore can display different and potent, I should say, activities toward different protein structures, different proteins that are not related to each other. For example, in this case, succinate dehydrogenase is not related at all to anything like cannabinoid system or protease of HIV, or for example TRPV1 protein of our sensory system. So based on these evidence, I should say that we should bear in our mind that traditional and conventional in silico, conventional chemistry approaches are far more ineffective than in silico and computational approaches, and this is one of the main reasons that many scientists are now drawing their attention to these newer and modern techniques to assess the ligand interaction with its protein partner and to enhance its effectiveness by modifying its structure. So this was the main point that I wanted to discuss with you in this series, and I hope you enjoy it. Be sure to subscribe to our channel, and thanks for watching.